Hello everyone, today we will be discussing about genetic manipulation of cells. You can see the whole procedure here in pictorial representation. Genetic manipulation is the modification of an organism's gene using technology. It is also known as genetic engineering and genetic modification. Genetic manipulation is a relatively new form of science. After all, the structure of DNA remained unknown until the work of Watson and Crick in 1953. The first recombinant DNA molecule was made by Paul Werg in 1972 by combining DNA from the monkey virus SV40 with the lambda virus. One of the first true genetic manipulation experiments took place 2 decades later, when biochemist Herbert Boyer and Stanley Cohen inserted DNA from one bacterium to another bacterium. In 1982, the FDA approved the first consumer GMO product, human insulin, to treat diabetes. Then method of genetic manipulation. First method is transgenic technology. This involves inserting genes from one organism into the DNA of another organism. The resulting organism is called a transgenic organism and it can express the inserted gene's traits. For example, genetically modified crops may be engineered to resist pest or product higher yields. Transgenic technology, also known as genetic engineering, is a biotechnology method that involves the introduction of genes from one organism into the DNA of another organism, resulting in a transgenic organism. This technology allows scientists to transfer specific traits or characteristics from one species to another, leading to expression of new genes or the modification of the existing ones. Then methods of transgenic technology. First one is the vector based transgenesis. In this approach, scientists use vectors such as plasmids or viruses to deliver the transgene into the host organism cell. The vector carries the foreign genes and helps it integrate into the host genome. Second one is the CRISPR-Cas9 and gene editing. While not traditional transgenesis, gene editing techniques like CRISPR-Cas9 can also be used to create transgenic organism by directly modifying the host organism's DNA. This allows for precise changes to be made in the genome. An application of transgenic technology in agriculture. We can see in agriculture, transgenic crops often referred to as genetically modified organism or GMOs have been developed to process traits such as pest resistant, herbicide tolerance, improved nutritional content and increased yield. Example include Bt cotton, resistant to certain pests, and genetically modified soya beans. In medicine, transgenic animals and cell cultures are used in biomedical research to study human diseases and develop treatments. Transgenic mice, for instance, are commonly used to model human genetic disorders. In biotechnology, transgenic microorganisms such as bacteria and yeast are used the production of pharmaceuticals, enzymes and other valuable bioproducts. These organisms can be engineered to produce specific proteins or chemicals. In conservation efforts, transgenics technology can be used to enhance the genetic diversity of endangered species or to introduce disease resistant into vulnerable populations. Ethical and environmental concern. Transgenic technology raises ethical concerns related to the environmental impact of GMOs, the potential for unintended consequences and issues of food safety and labeling. Additionally, there are ethical considerations when it comes to the genetic modifications of animals or humans. In regulation, the use of transgenic technology is subject to regulations and oversight in many countries. Regulatory agencies establish guidelines to assess the safety of transgenic organisms for human health and the environment. Future development. Advances in transgenic technology continues to occur, including more precise and efficient method of gene insertion and gene editing. Researchers are also exploring the potential for creating transgenic organism with novel capabilities and applications. Techniques like CRISPR-Cas9 allow the precise and targeted changes to an organism's DNA. This can include adding, deleting or modifying specific genes to achieve desired outcomes such as curing genetic diseases or improving traits in organism. In gene editing, that is very common and popular nowadays, also known as genomic editing, is a revolutionary biotechnological technique that allows scientists to precisely modify an organism's DNA, including adding, deleting or altering specific genes. This technology has a wide range of application in various fields, from medicine and agriculture to basic scientific research. Other methods like zinc finger nucleases, ZFNs and transcription activator like effector nucleases have also been used. In gene silencing, this involves inhibiting the expression of specific genes within an organism's DNA, which can be used to suppress certain traits or characteristics. That is, you can see in the pictorial graphics. Since the 1980s, genetic manipulation has been used in a variety of industries, including medicine, agriculture. Let us look into the few examples. In cancer treatment, in 2013, a gene editing tool called CRISPR was brought into life. It functions like a precious pair of scissors silencing pieces of DNA. Its first clinical trial in cancer therapy took place in 2019, making genetic changes to T cells, that is immune system cell, that in medicine production. Earlier we learned that the first GMO product approved was human insulin. Since then, genetic engineering has been used to mass produce a variety of drugs, including human growth hormone, albumin a chemical used in transport, anti-hemophilia drugs, infertility drugs and vaccine. Brewing yeast, a team of scientists in California have created a genetically modified yeast that mimics the flavor of a aroma of hops without any interaction with the plant. In agriculture, genetic manipulation is common in agriculture. Most GM crops were developed to prevent yield loss and control weeds. The three most common reasons for genetically manipulating crops are pest resistance, tolerance of herbicides used to control weeds and the resistance to plant viruses. It is not just a plant, genetic manipulation is used to transfer desirable traits between animals. Manipulation of genetic material in agriculture, selective breeding. Farmers have been using selective breeding for millennia to alter the traits of crops and livestock. How does it work? Farmers select individuals with desirable characteristics and breed them together. They choose the offspring with the best combination of traits and breed them together. This process continues over many generations until the trait has been established in the population. Using the selective breeding, farmers have converted wild plants and animals into the crops and livestock we use today. Desired characteristics include increased meat or milk production, larger fruit and eggs, increased nutritional content, disease resistant and calm temperature. Limitation of selective breeding. A trait can only be bred into individuals if it already exists in a species. So, selective breeding is limited by existing genetic variation. Selective breeding is a slow process, especially in animals. Offspring are genetically similar, so are equally vulnerable to the same diseases. Inbreeding can also result in a loss of alleles, limiting genetic variation. It is important to maintain genetic diversity in nature because new traits from wild plants can be bred into the crops to help them respond to climate change. Genetic diversity you can see in this picture. Then vegetative propagation. Vegetative propagation is a form of asexual reproduction. It involves taking part of a parent plant, causing it to regenerate into new plant. There are two methods, cutting and grafting. Cutting, a branch of the parent plant is cut off, its lower leaves removed and the stem is planted in compost. The plant hormones are added to encourage root growth. Plants grown by vegetative propagation are genetically identical to each other. So, useful characteristics will always be passed on. Vegetative propagation only required one plant and it is a very fast method of genetic manipulation. Then limitation of vegetative propagation. Artificially propagated plants have a lower lifespan than seed propagated plants. Like selective breeding, vegetative propagation is limited by existing genetic diversity. Vegetative propagation requires special training and it is more expensive than seed propagation. Genetically identical plants are equally vulnerable to the same diseases. Genetic manipulation ethics. Although GM crops and animal have excellent benefit. They bring a range of ethical concern. Some believes that it is not ethical to interfere with nature. Altering the genome of living organism in this way could be considered intrinsically wrong or immoral. There is a demand for transparent labeling guidelines, so consumer can make informed choices when purchasing GM food. Well-being concern. There are well-being concern of GM animals who cannot consent to genetic manipulation. Furthermore, selective breeding techniques regularly result in inbreeding. This increases the risk of inheriting harmful homozygous alleles that may cause disease or suffering in the animal. Financial control. GM crops are more expensive than regular crops. So, they can't be afforded by people in developing countries. In these countries, GM crop may add the reduction of poverty and improve food security. High prices are linked to GM crop parity. Farmers are not allowed to save GM seeds for the next season, but must instead buy the new each year at a set price. Designer babies and eugenics. If we can manipulate the genetics of animal, why could not we do the same to our slave? We can to a minor extent. Couples who are caring for a genetic disease can undergo pre-implantation genetic testing. Embryo fertilized via IVF are tested for a range of genetic disorders before being implanted into the woman. Although it helps prevent lifelong disease and disability, pre-implantation genetic testing has raised ethical concern. What if manipulating genetics went further? Experts have warned about the dystopian future of designer babies and it links to human eugenics. Eugenics is the practice of altering a population's genetic composition through selective breeding and forced sterilization. If parents can pick and choose traits of their future offspring, ranging from their eye color to their personality and intelligence, just imagine what would be happen to the society. So, the advantage of the genetic manipulation for preventing diseases and pests, resulting in reduced demand for chemical pesticide and insecticide, then enhanced nutritional content, then faster growth rate, increased yield, can survive in a wider range of climatic conditions, and the disadvantages are high cost, may take several generations, require specialized workers and equipment, ethical concerns, risk of ecological imbalances leading to diseases, allergens and toxins, may lead to reduced genetic diversity. Now, the general steps involved in genetic manipulations. First, we have to identify the target gene or DNA sequence. The first step in genetic manipulation is to identify the specific gene or DNA sequence that you want to modify or introduce into the host organism. This could be a gene responsible for a desired trait or a sequence associated with a particular function. Then next point is select the host organism. How we select the host organism? Choose the organism in which you want to make the genetic modification. It could be a plant, animal, microorganism or cell culture. The choice of host organism depend on the intended application. Third one is design the genetic modification. Plant and genetic modification by determining what changes need to be made to the target gene or DNA sequence. Decide whether you want to add, delete or modify specific genetic elements. Then choose a genetic or gene editing tools. Select the appropriate gene editing tool or technique for your project. Common gene editing tool include CRISPR-Cas9, zinc finger nucleases, transcription activator like effector nucleases or other specialized methods. Design guide RNAs or DNA binding proteins. If using CRISPR-Cas9 or similar system, design guide RNAs or gRNAs that are complementary to the target DNA sequence. In the case of ZFNS or TALNS, design DNA binding protein that can recognize and bind to the target sequence. Then delivery of editing components. Introduce the gene editing component, for example, CRISPR-Cas9 or gRNAs or DNA binding proteins into the host organism cells. This can be done using various methods including electroporation, virus vector, microinjection or other delivery techniques. Edit the DNA. How we edit the DNA? So, allow the gene editing tool to act on the target DNA within the host cell. For example, CRISPR-Cas9 will cut the DNA at the desired location, creating a double standard break. Then repair mechanism. The host organism cellular repair mechanisms will attempt to fix the DNA break. Depending on the desired outcome, you may rely on two main repair pathways, non-homologous and joining NHEJ. This often results insertion or deletion at the site of the break, potentially disrupting the target gene. Homology directed repair. If a repair template with the desired genetic sequence is provided, the cell may use it to repair the break, leading to precise gene editing. Then select and isolate modified cell. Identify and isolate the cells or organism that have undergone the desired genetic modifications. This can involve selecting for cells with specific traits or using markers to identify edited cells. Then characterize and validate the modification. Confirm that genetic modification has been successfully carried out. This may involve DNA sequencing, PCR analysis or functional assay to assess the desired trait or gene expression. For propagation and breeding for multicellular organism, if working with multicellular organism such as plants or animals, propagate or breed individuals that carry the desired genetic modifications. In regulatory compliances, ensure compliance with any regulatory requirements or ethical considerations associated with your genetic manipulation work. This includes adhering to safety protocols and guidelines. So, the genetic manipulation is a complex and multifaceted field that involved a wide range of factors including scientific, ethical, regulatory and practical consideration. In scientific factors, targeted gene selection, Choosing the right gene or DNA sequence to manipulate it's crucial, whether it's for therapeutic purpose or agriculture improvement or scientific research. Choice of gene editing tools, selecting the appropriate gene editing techniques such as CRISPR-Cas9, ZFNS or TALENs depend on the specific project goals and the organism being modified. Precision and accuracy, ensuring the accuracy and specificity of the genetic modification is critical to avoid unintended consequences. Ethical and moral consideration, human germline editing gene in human germline cell, for example, sperm, eggs or embryos, raises significant ethical questions about designer babies and long term consequences for further generations. Environmental impact in agriculture, concern about the environmental impact of genetically modified organisms relate to issues like gene flow in wild populations and potential ecological disruption. Consent and in autonomy, in this consent human gene editing for therapeutic purposes, issues related to informed consent and individual autonomy must be addressed. Regulatory and legal frameworks. Government regulation, many countries have established regulatory agencies and frameworks to oversee genetic manipulation, ensuring the safety of both humans and the environment. In compliances, researchers and organizers must adhere to applicable laws and regulations, which may vary by jurisdiction. In safety protocol, containment, proper containment measure must be placed to prevent the accidental release of genetically modified organism into the environment. In biosecurity, ensuring that sensitive genetic information or materials are protected, practical and the technical consideration, delivery method, selecting the right method for introducing gene editing components into target cells or organism is essential. Validation and testing, comprehensive testing and validation of gene modifications are necessary to confirm the intended outcomes. In intellectual property and ownership, patents and licensing, the ownership of intellectual property related to the genetic manipulation techniques and the application can have economic and innovation implications. In research and development, funding, adequate funding and resources are essential for advancing genetic manipulation technologies and application. In collaboration among researchers and institutions can accelerate progress and improve safety standards. In summary, genetic manipulation, also known as genetic engineering or genetic modification, is the process of altering the genetic material, that is DNA, of any organism to achieve a specific outcome. This manipulation can serve various purposes such as improving crop yields, creating genetically modified organism or GMOs, studying gene function or developing gene therapies for medical conditions. Several important factors surrounding genetic manipulation, including the purpose, technique used, ethical consideration, regulations, safety, environmental impact, long-term effect, social acceptance, intellectual property, scientific advancement, collaborations, transparency and biosecurity. striking a balance between these factors is essential for responsible and ethical application of genetic engineering technologies. Nowadays, the genetic manipulation in cell is a very important, because the targeted gene therapy is very advanced and we have to culture the cell first, and their different factors are there to generate the cell. So, we have to target to generate the genetic manipulation of cell, and we have to do work for that. So many advances are there, so many scopes are there for the future. So, our student go for that and they have to work on that. Okay? Thank you very much.